Hi everyone. It's great being here, and I would like to thank the organizers for inviting me. So Yoni, Rami, and Mario. Um, okay, so yes, as Yoni mentioned, I'm from the Faculty of Medicine, working together with Yoni. And actually, I'm not from the. You probably know I'm not from the addiction field. I come from vision, the visual system. But recently we started also looking at effects of light on addiction. Okay, so after the last 2 days, probably all of you know how the prefrontal cortex, nucleus accumbens, VTA, and other parts of the reward system are connected. However, today, thank you. However, today I'll give you a small but important update. So in 2018, the lab of Samer Attar from NIH discovered that the prefrontal cortex and nucleus accumbens receive input from a small dorsothalamic nucleus here that borders the lateral habenula. In a burst of creativity, they called it the perihabenular nucleus, or PHB. This nucleus apparently receive input from the retina, and specifically from intrinsically photosensitive retinal ganglion cells, or IPRGCs. Okay, these cells continue to respond to light for many minutes or even hours, and these cells also encode the intensity of light. So in essence, they act as a light meter. And so this raises the possibility that perhaps light through this pathway of IPRGC to the PHB can modulate prefrontal and accumbal activity and therefore modulate reward processing. So to find out, we systematically studied the effect of light along these pathways, and we started with the prefrontal cortex. This work, most of this work, was led by a super MD PhD student, Eleshiv Tsangen, with the help of many others that you can see here. The core experiment was extracellular recordings from awake, age-restrained mice while shining on their eyes light at 7 different light intensities. Okay, so in the prefrontal cortex, in total, we recorded from 20 mice in which we identified over 1,600 neurons. 43% and 35% of these cells showed a significant response to light, either transient response or a persistent response. These light responsive neurons were located throughout the medial prefrontal cortex, including in the anterior cingulate, prelimbic, infralimbic cortices, and also in the dorsal taenia tecta, in the dorsal peduncular area, and the dorsal taenia tecta. Can you hear me? It's okay? Okay. Okay, so we were worried that perhaps recording from any brain region would identify such light-responsive neurons. This, however, was not the case. Recording from the nearby secondary motor cortex show that the incidence of transient and persistent light-responsive neurons was significantly lower in the secondary motor cortex than in the prefrontal cortex. In addition to light responsiveness, we wanted to know if these neurons can encode the intensity of light. To find out, we extracted the steady-state firing rate across the 7 light intensities, feed them to a sigmoid, and then cells with an RMSE fit below the 5th percentile of a null distribution were labeled as intensity encoding. In total, 15% of all identified neurons were found to be intensity encodings, and these neurons were mostly located in the prelimbic and infralimbic cortices. we discovered 4 different response types of these persistent intensity encoding neurons. 2 types here in blue, in red and blue, transiently increase their firing rate, both when the light went on and when it went off, but otherwise either increased or decreased their steady state firing rate. And cells of the 2 other types slowly increased or decreased their firing rate. The steady state firing of the 2 enhanced types in red and magenta monotonically increased with light intensity, while the steady state firing rate of the 2 suppressed types in blue and green monotonically decreased their firing rate with increasing light intensity. Okay, so these neurons can encode the intensity of light in response to discrete steps of intensity. To find out whether these neurons can also track or continuously modulate their firing rate in response to intensity gradients, we use the biphasic stimulus, in which log light intensity was linearly increased and then decreased over 60 seconds. Here in the 2 enhanced types in red and magenta, firing rate slowly and gradually increased and then decreased, while in the 2 suppressed types in blue and green, these types showed the inverse response. So these neurons can also closely track the intensity of light. Now, interestingly, light affected different subregions of the prefrontal cortex in a different manner. Most of the infralimbic neurons, most neurons in the infralimbic cortex were enhanced by light, while most neurons in the prelimbic cortex were suppressed by light. Consequently, the average firing rate across all neurons was above baseline in the infralimbic cortex, but below baseline in the prelimbic cortex. And when looking only at intensity encoding neurons, we can see here that the firing rate in the infralimbic cortex was significantly higher than in the prelimbic cortex, and both sat on both sides of the baseline. Now, this dichotomy mirrors the PL-GO, IL-stop model that holds that the PL is active during execution or expression, while the infralimbic is active during inhibition or extinction. Therefore, these results suggest that the possibility that light may inhibit drug seeking by suppressing prelimbic activity and simultaneously increase excitation, or and simultaneously facilitate extinction by enhancing infralimbic activity. Okay, and we will get back to this point later. Okay, so next we wanted, we hypothesized that prefrontal sensitivity depends on input from the PHB and also on signals from IPRGCs. To find out, we in OPN4 Cree mice, we expressed the inhibiting GI dread, specifically in IPRGCs, and then recorded prefrontal activity following CNO or saline infusion. Control mice expressed only M-cherry. So here we can see that CNO significantly reduced the firing rate compared to saline in dread mice, but not in control M-cherry mice. And when we look here at all 7 intensities, so let's focus first on the thick orange line here. So these are dread mice receiving saline. So we can see strong dependence on light intensity, but this dependence weakens dramatically after adding CNO here, the thick gray line. Okay, and the saline CNO differences were significant at the 4 highest intensities here. On the other end, in M-cherry mice that are represented here by thin lines, the differences between CNO and saline were much smaller. So based on these results, so we can say that the reduction in firing rate in prefrontal cortex firing rate is primarily due to inhibition of IPRGCs using DREAD, but there's also a modest component of DREAD-independent CNO effect. Now, to further substantiate this or our claim of the dependence of prefrontal cortex activity sensitivity on IPRGCs, we ablated IPRGCs using diphtheria toxin A, DTA. Here you can see that DTA mice, here in gray, showed significantly lower incidence of responsive and intensity encoding neurons compared to wild-type and M-cherry mice that are shown here in blue and orange. And if we look at all 7 intensities, so you can see that M-cherry and wild-type mice are almost identical and show strong dependence on light intensity, while DTA mice show much weaker dependence. Okay, so based on this experiment and also based on the chemogenetic inhibition from last slide, we can say that prefrontal sensitivity strongly depends on IPRGC input, but still we cannot exclude, because there is some residual here, residual responses, so we cannot exclude, of course, incomplete expression of viruses. And on the other end, maybe additional inputs or additional sources of light sensitivity in addition to IPRGCs. Then we went along the pathway and tested the dependence of prefrontal sensitivity on input from the PHB. Very short, we used chemogenetic inhibition and also optogenetic inhibition, and both approaches showed that prefrontal light sensitivity depends on PHB input. Okay, so to summarize this part, we identified so 15% of prefrontal neurons persistently encode light intensity. These neurons are divisible into 4 different subtypes. Light responsiveness and intensity encoding depend on IPRGC input and also on PHB input. And due to the opposite effect of light on prelimbic and infralimbic activity, light may inhibit drug seeking by suppressing prelimbic activity, and on the other end, promote extinction learning by enhancing infralimbic activity. All right, so that was about the prefrontal cortex. Now let's move to the nucleus accumbens, and these data have not been published yet. So here we recorded from 21 mice in which we identified over 1,000 neurons. We identified neurons both in the core but also in the shell. And here, persistent light responsive neurons accounted for 45% of the identified neurons, even more than in the prefrontal cortex. And persistent intensity encoding neurons were 1/3 of all neurons. So that's a lot. 1/3 of neurons recorded from the nucleus accumbens could encode the intensity of light. Here we identified 3 different response types, 2 in which steady state firing increased or decreased with a small off response, and a 3rd type with transient on and off responses and very modest suppression of steady state firing. The steady state firing in all 3 types varied monotonically with light intensity. And as we did for the prefrontal cortex, also accumbal neurons could track closely the intensity of light. Now, so we found that light had almost opposite effects on the core and the shell. Okay, so most core neurons were enhanced by light, while most shell neurons were suppressed by light. And this is evident also here. So this is the mean firing rate. So we can see that light enhances firing rate in the core, but suppresses it in the shell. And here we can see that both core and shell can encode intensity, but with opposite slopes. All right? And then we moved on to, sorry. Yeah, and then we moved on to, we wanted to determine whether these light intensity encoding neurons are D1 or D2 MSNs. To this end, we used optotagging. We expressed channel rhodopsin either in DRD1 cremase for D1 cells or in A28 cremase for D2 cells. And here we can see that D1 cells were actually a mixture of both enhanced and suppressed types, while D2 cells were only enhanced cells. And this is also evident here. So this is just the firing rate. We can see that the firing rate of D1 cells is centered around 0 and is significantly lower than in D2 cells. Next, as we did for the prefrontal cortex, also here we used optogenetic inhibition of PHB input and terminal ablation of IPRGCs, and we found that acamble light sensitivity depends on input from IPRGCs and the PHB. And again, also in this case, there is some residual response, so we cannot exclude any the possibility that there are additional sources. Okay, so to summarize this part, so a third of acamble neurons persistently encode light intensity. They are divisible into 3 different types. We found that opposite effects of light on the core and shell. Now, the suppression of the shell may reflect glutamatergic transmission from the PHB to FSIs, to fast spiking interneurons, which would lead to feed-forward inhibition on MSNs. Or it could also reflect GABAergic transmission from the PHB directly on MSNs. We also found that light bidirectionally modulates D1 cells but selectively enhances D2 cells. And this leads, this only in the core, by the way, and this would lead to a net increase in core activity, which is exactly what we found here. It would also say that, or suggest that light may inhibit drug preference and seeking, both by suppressing the nucleus accumbens shell and enhancing its core. And finally, I don't know why it's cut here, but we found that we show that light sensitivity in the nucleus accumbens depends on both PHB input and IPRGCs. Okay, so we know that the prefrontal cortex and nucleus accumbens encode light intensity. We also know that these effects depend on IPRGC input. We also found contrasting effects in the cortex between the IL and the PL, and also between the core and the shell. And this naturally leads to the question, can light exposure reduce preference, drug preference, by modulating prefrontal and accumbal activity? Something strange here in the presentation, never mind. So to this end, so we used the condition, the cocaine condition place preference. This work was performed in collaboration with Yoni Kupchik, the lab of Yoni Kupchik, and was led by Nuran Abed El Haq and Diti Inbar. So the CPP chamber included 2 halves, one with black and white stripes and the other being uniform gray. The CPP protocol included handling, habituation, and then conditioning, in which mice were injected with cocaine or saline and were confined to a specific half of the chamber. And then we had abstinence, and then the first test, in the CPP test, and it was performed under either bright light, 3000 lux, or dim light. Okay? And then we had another phase of conditioning and abstinence and another test, but now under the other light condition. So each mouse served as its own control. Each mouse was tested both under dim and under bright light. All right, so these are the results. So following dim light conditioning, bright light during CPP test decreased the CPP score compared to dim light. Okay, these are results for 12 mice, but here you can see an example in one of the mice, dim, bright. This is the CPP score. Okay, so now if this CPP decrease under bright light were an effect of a context shift, we would expect that following bright light conditioning, CPP would be actually lower under dim light, but this was not the case. Following bright conditioning, the CPP score was similar under both bright and dim light conditions. Therefore, bright light reduces cocaine preference, and it is not, and it's probably not an effect of context shift. Now, this reduction of CPP under bright light disappeared after ablating IPRGCs in OPN4 Cre mice, but it stayed intact in control mice that expressed only M-cherry. So this shows that light modulates cocaine preference via an IPRGC-driven light sensitivity, likely through modulation of the prefrontal cortex or the nucleus accumbens, and that's exactly what we are looking at now. All right, so now let's try to see how all our results fit together. Bright light during CPP testing engages the IPRGC and PHB pathway. It enhances infralimbic activity but suppresses prelimbic activity. These two together push the prefrontal cortex toward a stop signal, which would lead to a reduction in CPP expression. In parallel, as we saw, light also suppresses shell activity, and this would further weaken or decrease CPP expression, and that's exactly what we see in our behavioral results. Okay, so until here, so that's what we have shown so far, but we also found that light enhances core activity. Now, this dual shift of the shell and core may potentially also lead to cue-induced seeking, also to a reduction in context and drug priming, and therefore a reduction in relapse, and on the other end may increase extinction learning. Okay, and extinction learning is actually also further aided by this enhancement of infralimbic activity. So we plan to look at the effect of light on cue seeking, relapse, and extinction. And actually, we've started in humans looking at the effect of light on craving and extinction. And so if indeed light affects at least some of these phases or stages of addiction, we hope that bright light therapy, which is scalable and very low cost, will be able to ultimately be integrated as standard addiction care and to acutely reduce drug seeking and relapse, and on the other end to boost extinction learning. And with that, I would like to thank my lab members, my collaborators, and thank you for listening.